Hey everyone, and welcome to this explainer. Today, we're doing something really special. We're not just, reading through some dry technical manual. We are embarking on an epic human quest to map the invisible frontier. We're going to trace the entire history and evolution of the microscope and look at exactly how humanity managed to expand our vision into a completely unseen world.
Okay, let's dive right in. To make things easy, we've broken our mental roadmap down into three main chunks. First up is the quest, which is our historical journey. Then, the language of the lens, where we'll cover the essential optical vocabulary you really need to know. And finally, the modern arsenal, where we explore the incredible tools that literally let us look at individual atoms.
So, Act 1, the quest. Every great invention usually starts with a frustrating physical limitation. And for us, it's the simple biological fact that the naked human eye just can't distinguish objects that are smaller than about 0.1 millimeters. That hard biological barrier, that is exactly what sparked the creation of the microscope in the first place.
It's amazing to think we can cover 400 years of history in one quick sweep. We start way back in the 1590s with Zacharias Janssen and his kind of rudimentary brass tubes that gave you maybe 3 to 9 times magnification. From there, we jump to Antony von Leeuwenhoek actually discovering microbes in the 1670s. Then, a massive leap forward to Ernst Röska's electron breakthrough in 1931. And finally, we arrive at Gerd Binnig and Heinrich Rohrer literally manipulating atoms by 1981.
Now think about Antony von Leeuwenhoek's single-lens design for a second. It was this tiny, almost basic-looking little device, but it was an absolute game-changer. Why? Because it actually bypassed the severe optical distortion that plagued early compound lenses. By crafting this near-perfect spherical lens, he achieved a staggering 270 times magnification. No wonder he used it to discover the microbial world, and rightfully earned the title Father of Microbiology.
All right, moving right along to Act 2, the language of the lens. Before we get into the really modern tech, let's do a quick training sequence to learn the optical cheat codes that determine how a microscope actually performs.
First up is resolution. By definition, this is the minimum distance required to distinguish two adjacent points as separate independent entities. Think of it exactly like the pixels on your smartphone screen. A smaller numerical value for that distance means you can see finer, much closer details without them just blurring together into one big blob. Now, a concept that's absolutely crucial here is numerical aperture, or NA. It's a dimensionless number that basically represents how wide of an angle a lens can gather light from. A higher NA means you're capturing more light and finer diffraction details. And actually, we can even boost this capability by putting a special high refractive oil right between the lens and the sample. Pretty clever, right?
Now, I will say, it is incredibly easy to mix these two terms up when you're reading the research. So just remember this. Resolving power is the overarching, broad capability of your optical system to distinguish points. Resolution, on the other hand, is the strict, specific mathematical distance metric of that capability.
Of course, lenses aren't perfect, unfortunately. They suffer from what we call aberration, basically optical defects that cause blur or distortion. The two main culprits are chromatic aberration, which causes color fringing because different wavelengths focus at different points, and spherical aberration, where the center and the edges of the lens focus differently. Modern microscopes actually use these incredibly complex lens combinations just to correct those exact color and shape errors.
Which brings us to Act 3, the modern arsenal. Let's take a look at how humanity built highly specialized tools to completely bypass physical limitations, like Ernst Abbe's optical diffraction barrier, for instance.
So when we talk about phase contrast in dark field techniques, here is why this actually matters to you. Phase contrast takes invisible shifts in light waves and actually converts them into visible brightness differences. This is a huge deal because it allows us to observe living cells in real time, completely bypassing the need to kill them with toxic dyes just to see what their structure looks like.
And that leads us to the two absolute giants of electron microscopy. First, we have TEM, or transmission electron microscopy. This shoots electrons straight through ultra-thin slices of a sample to give us a highly detailed, two-dimensional view of internal structures. Then we have SEM, or scanning electron microscopy, which scans the surface to generate these incredibly rich, three-dimensional textures.
When you compare these mechanics side by side, it's wild. Ruska's TEM completely shattered the visible light barrier, achieving astonishing sub-nanometer resolution so we could see deep inside cells. Meanwhile, Manfred von Ardenn's SEM concept gave us such a massive depth of field that everyday microscopic surfaces suddenly looked like incredibly detailed miniature sculptures.
Let's move on and see how all this builds into the late 20th century, because eventually humanity moved beyond lenses and beams entirely. We leaped straight into the quantum realm with scanning probe microscopes. We started using actual physical probes to literally touch the atomic world.
The way a scanning tunneling microscope, or STM, works is honestly mind-blowing. By bringing a conductive probe incredibly close to a sample, it measures the quantum tunneling current. It is exactly like feeling the shape of a tiny object in absolute darkness using an incredibly sharp needle. This technique is so precise, it even allowed scientists to literally push individual atoms around.
But STM does have a bit of a flaw. It requires conductive materials. While the atomic force microscope elegantly overcomes this by measuring physical van der Waals forces using a tiny bending cantilever. This is huge because it means we can finally examine insulators, proteins, and living cells at the atomic level even while they are completely submerged in liquid. So we've journeyed all the way from Zacharias Jansen's simple brass tubes to physically mapping and manipulating the very building blocks of the universe, which leaves us with one highly provocative question. Having already reached the atoms themselves, what invisible frontier will humanity uncover next? Thanks so much for joining me for this explainer.